Hello everyone, my name is Giulia Capestro and I'm here with my colleague Silvia Zaoli. We are data scientists in Novartis and today we are going to talk to you about a company internal data challenge in subgroup identification. So let's first understand what is subgroup identification and why we need it. Let's consider the following situation. We design and run a phase 3 clinical trial. A phase 3 trial aims to test the efficacy of a certain treatment and compare it to a treatment already approved from the authority, called standard of care. We then submit our results to the regulatory agency, but we do not get approval because the efficacy is inadequate compared to the standard of care. So what do we do now? The situation is pretty common, and according to a study published from the Journal of the American Medical Association, 50% of drugs failed their first submission. 13% of them is rejected because their efficacy is inadequate compared to the standard of care. And this percentage increased to 60% of drugs that will never be approved. However, the fact that the efficacy is inadequate on the trial population doesn't mean that the drug is useless. But patients are heterogeneous, and so they respond to the drug. Patients are characterized by baseline covariates or biomarkers, such as demographic, disease subtypes, severity scores, genetic biomarkers, and so on. Covariates can be prognostic and predictive. Prognostics inform about the patient's outcome regardless of the treatment received. So, for example, a younger patient may recover more likely from a disease regardless of the treatment. And in this case, age is the prognostic covariate. Predictive covariates inform about the effect of the treatment. And for example, if the treatment has more chance to work on a patient that have certain symptoms, symptoms is a predictive covariate. So if we can find out which covariates are predictive, we can identify a subgroup with increased treatment effect. A subset of patients defined by predictive covariate where the treatment effect is higher than its complement. An example could be patients younger than 60 that have a symptom X. It's important to remark the difference between subgroup identification and clustering. In clustering, we try to divide patients based on their similar characteristics. Subgroup identification, we try to find a subset where the treatment effect differs from its complement. So the task of subgroup identification is strictly connected to precision medicine. In fact, one of the aims of precision medicine is to find which drug works best for a patient with certain characteristics. Coming back to our motivating example of a failed clinical trial, if we manage to find a subgroup with the increased treatment effect, we can then plan a new follow-up trial with the new inclusion criteria. However, identifying a subgroup that will have increased treatment effect in a future trial is a hard problem. And in fact, the risk is that the prediction will not replicate in the follow-up trial. The treatment effect is also could be noisy due to small sample size. And the original trial we need to think that it was designed to find a treatment in the wider population and not in a subgroup. A funny example of this is a trial on the efficacy of the aspirin, where it came out that the drug was harmful for people born under Libra and Gemini. So for this reason, the problems still remain challenging, even if we have the most reliable methods. Here are the most common methods to solve this problem. We have tree-based and regression-based models. In tree-based, we have the decision tree that split patients based on their characteristics. The criteria differ from method by method, and the model returns a combination of threshold which define the subgroup. The regression-based model identifies predictive variables thanks to the regression on the outcome, but does not directly return subgroup definition. So we are often in this situation where we have to decide whether to design or not a follow-up trial. Therefore, we wanted to test how good are our team at subgroup identification. To answer this question, we designed a challenge where the participant faced a realistic situation and their solution was tested on unseen data. The challenge was also an opportunity to experience a new way of working and compare methods without having the pressure of a real project. Finally, the challenge was hosted on the Novartis Data42 platform, which stores a large amount of clinical and real-world evidence data, where users can choose to use several different data analysis tools, and where we foster collaboration and reproducibility. So now my colleague Silvia will introduce to you the challenge data.
Thank you, Giulia. So, as Giulia mentioned, we wanted the challenge to reproduce a realistic situation where the participants could identify the subgroup in past clinical data, but then their solution would be tested on new unseen data. So this situation would allow to test the reproducibility of the identified subgroup in future trials. And to this aim, we structured the challenge data into a training and a scoring dataset. The training dataset was the one that was provided to all the participants, and it consisted in 4 phase 3 clinical trials, randomized clinical trials, in the same therapeutic area. Then the solutions that were found by the participants based on this training dataset were scored by us on one more clinical trial, also in the same therapeutic area, that was not available to the participants. All the clinical trials had the same inclusion criteria, and for each patient, more than 90 covariates were available. As an endpoint for the challenge, we considered a binary response index, which is a combination of several measures of patient improvement, including, for example, a patient global assessment, physician global assessment, the results of several laboratory tests, etc. And based on this binary response, we then define the treatment effect as the difference between the probability to respond under treatment and the probability to respond under control. The participants had to submit 3 things. First of all, they had to submit a definition of the subgroup for which they predict an increased treatment effect in the new trial. An example of such definition could be age smaller than 60. Then they had to submit their prediction for the magnitude of the treatment effect to be observed for the subgroup that they proposed in the new trial, which we call a delta pred. And also they had to submit an estimation of the uncertainty for that prediction that they're giving, which we call sigma pred. And finally, they had to submit a description of their methodology, including a clinical or biological justification of the subgroup that they are proposing. We evaluated the submissions based on 2 criteria. The first criterion evaluates if the subgroup proposed by the team has indeed an increased treatment effect in the new trial. To evaluate this, we modeled the probability of a patient's eye to respond as a logistic regression with a term that depends on the treatment allocation, a term that depends on the subgroup belonging, and a term that depends on their interaction, the interaction of the treatment and the subgroup belonging. So if the coefficient for this interaction term, beta interaction, is positive, this means that the subgroup has an increased treatment effect in the new trial. And therefore, we defined our first score as a test statistic for this interaction coefficient. The second criteria that we used evaluates how accurate is the prediction of treatment effect that the participants proposed. In the new trial, we can measure what is the observed treatment effect for that subgroup, and we call this measure delta hat. Then the participants proposed instead, they submitted their prediction for this value, which is delta pred. So our second score is the log likelihood of the observed value delta hat according to a Gaussian distribution centered around their prediction delta pred and with a standard deviation equal to the uncertainty that they've submitted sigma pred. Okay, let's now have a look at how the subgroups that the different teams submitted look like. So first of all, you can see on the plot here on the left that typically few variables, few covariates were used in the definition of the subgroups, although there is a bit of variability. They range from 1 to 8. And there are a few variables that were used very often in these definitions. For example, age was appearing very often in the subgroup definition. And this could suggest that the subgroup proposed by different teams were relatively similar. This is actually not the case, as you can see from the plot on the left. In this plot, each dot is a team, and two dots are close to each other if the subgroups that the two teams proposed are similar according to a Jacquard similarity index. And instead, they are far from each other if the subgroups that they proposed are different. And you can see that the three top teams, these teams numbered 1, 2, 3, these are the teams that scored the best, and they are relatively far from each other. So this means that the three best teams submitted subgroups that were relatively different. And what this means is that there is not an obvious solution for this subgroup question. There's not an obvious subgroup, but different possible solutions. Almost all the teams managed to identify a subgroup that has an increased treatment effect with respect to the overall one in the new trial, as you can see from the plot on the left. Here, this continuous line is the overall treatment effect in the new trial, and the dots are the treatment effect in the subgroups proposed by the different teams. This suggests that there is hope to give an answer to the subgroup identification question. However, there is something that I would like to remark, which is that the teams systematically overestimated the treatment effect. This means that the value that they predicted for the treatment effect in their subgroup was almost always larger than the value that was observed in the new trial. This is a known statistical effect, which is called regression to the mean, but still it's important to keep it in mind in case we are considering whether to make a follow-up trial. In terms of methods that were used by the participants, if we focus on the top teams, two of them used the penalized regression methods. In one case, a lasso regression, and in another case, a dose-response model with shrinkage prior. This method allowed them to identify which of the covariates are predictive of the outcome. But then to arrive to a subgroup definition, they still had to find thresholds for those predictive covariates. And they did that by trying different combinations of the thresholds and evaluating which one worked best by bootstrap. Another team instead used a tree-based model called MOB, and this allowed them to directly arrive to the subgroup definition. To conclude, I would like to tell you about what we learned from this challenge. First of all, the challenge confirmed that subgroup identification is really a hard problem. In fact, no obvious solution emerged, both in terms of the subgroup. In fact, different teams found different subgroups that all had an increased treatment effect, and also in terms of methods, because the different teams that managed to find good solution also used relatively different methods. And this is an especially interesting outcome of this kind of challenge, because in a known challenge situation, you would typically only have one team tackling this issue and coming up with one solution, And so maybe you would not realize how many different alternative good solutions exist. Also, all the participants highlighted how tough it is to evaluate whether your subgroup will replicate in a new trial. To evaluate that, they resorted to a mixture of statistical and clinical insights. From the statistical point of view, they used the cross-validation, holding out, for example, each trial in turn, and so they could identify the subgroup on the training trials and then evaluate it on the held-out trial, so that they would find a subgroup that not only has a high treatment effect, but also whose treatment effect is consistent across the held-out trials. And so it has a higher probability to replicate in a future trial. From the clinical point of view, the participants favored subgroups based on covariates that had a clinical meaning. And to evaluate that, they had the possibility to consult with clinicians during the challenge. Related to this last point, you might remember that at the beginning, Giulia said that data from previous clinical trials were not meant statistically to answer to the question of subgroup identification. They were meant to measure a treatment effect on the whole population, not on the subgroup. So this means that in this case, the data could statistically not be sufficient to give a significant answer to this question. It's a situation where the data do not speak for themselves. And so even if we have the best method, the most powerful method, there might be the need to complement data with external information, external to our data set, which might be, for example, a clinical insights or information from preclinical studies. And with this, I would like to conclude and to thank the other organizers of the challenge, Bjorn, Karsten, Connor, Mark, Michaela, Ruvi, and Jana, the participants of the challenge, Data42 that allows us to be here to speak, and all of you for listening. Thank you.